Hello again, students. So now that we understand the mechanics of how cells actually divide, we're going to take a look at how that division is regulated. Um, so the importance of this, I think, is highlighted by what happens when cell cycle regulation is not happening. So if we don't regulate when cells divide, they begin to divide uncontrollably. And that results in a condition that I'm pretty confident every one of you has heard of. Do we know what it is? It is cancer. So cancer is uncontrolled cell division. And unfortunately, it is quite likely that if you don't now, at some point in your life, you or someone you know is going to end up with cancer. It affects lots and lots of people, and so it's worth understanding how this works. So one word you probably hear associated with cancer is the word tumor. And what a tumor is it's a mass of undifferentiated cells. In other words, the cells are dividing uncontrollably, and as they divide uncontrollably, they become this mass. And that mass can manifest itself as this like physical bump. And there's different types of cancers depending on which cells are being affected. And you might have, so the bumps could look like this in skin cancer. It could be a little tumor in lung cancer. It could be little tumors in your colon for colon cancer, tumor in the breast tissue for breast cancer. So what they all have in common is there's these cells that are not behaving properly. They're dividing uncontrollably and then forming this mass. Now, there are different types of tumors, and two broad categories are benign or malignant. So a benign tumor means that the cells are remaining at their point of origin. And so here you have a benign, non-cancerous tumor, which basically means they're just sort of forming and they just kind of form this little lump, but they're not really spreading at all from that spot. A malignant tumor, on the other hand, is much more dangerous because here the cells are spreading and invading into other tissues. And that process is called metastasis. So metastasis is the spread of cancer cells into the bloodstream and to other cells. There's a nice animation here. You need a flash player to watch it, unfortunately. But I'll kind of go through in this diagram in case you can't access that. And what happens is as the tumor grows, these cells become more as it grows, then the cells sort of eventually begin to invade the underlying tissue. And if the cell and the tumor gets close to the bloodstream, those cells can actually enter the bloodstream. And then from the bloodstream, it can actually spread to other parts of the body. And so you might think, well, skin cancer, okay, so I have a big lump of cells on my skin. Who cares? I'm not going to die because I have a lump of cells on my skin. But the reason why skin cancer is so dangerous is because that lump of cells in your skin could enter your bloodstream and spread to other parts of your body where they could interfere with the organ function and cause some significant issues. So what do we do to treat cancer? I really, really urge you to click on that link and watch this TED Talk because it does a really great job talking about it. You may have heard of chemotherapy and radiation therapy, and what both of these things have in common is they're used to treat cancer. Now, the way that they both work is they attack cells while they're dividing. So, for example, one type of chemotherapy drug actually damages the spindle, which prevents a cell from dividing. And that's good if you're trying to kill cancer because the cancer cells are dividing uncontrollably. So if you attack the cell as it's trying to divide, those cells that are dividing constantly are going to struggle and be killed off. The problem, though, is that those same drugs are also going to affect normal cells as they try to divide. And cells in your body that divide frequently are your hair cells. And so that's why people with chemotherapy, undergoing chemotherapy, lose their hair because those hair cells are being attacked and killed along with the cancer cells. Another symptom is nausea and vomiting. You just feel sick to your stomach, and that's because the lining of your digestive tract divides quite frequently because food is constantly passing through and cells are getting damaged as it happens. So normally your digestive system is continually regenerating cells. Well, chemotherapy destroys those cells, and so then your digestive tract can't repair itself as well, and so you feel sick to your stomach and nauseous. So, unfortunately, these drugs are the best tool we have to deal with the cancer, but they do cause these unfortunate side effects. Radiation therapy, similarly, they radiate hopefully a targeted spot, which can target the cells, but again, that can cause some damage as well. So this TED Talk explains it well. I encourage you to watch it. So then, how then do we regulate cell division to prevent cancer? Well, normally what we have is there's a couple ways that we regulate this under normal circumstances. One is what's called a cell cycle clock. So this refers to rhythmic fluctuations in cell cycle control molecules, and these will regulate the timing of the cell cycle. So what does that mean? Well, cells have like a natural clock. Like skin cells are supposed to divide every so often. Stomach cells divide every so often. The lining of your blood vessel cells divide every so often. And so it's specific to each cell how often that happens, but each cell has this little clock inside it. And so you might wonder, well, how does that work? How does a cell keep time? And the way they keep time are with these molecules called cyclins. And cyclins are just proteins, but their levels fluctuate during the cell cycle. So they're kind of produced, they spike, and then they're broken down. Produce, spike, broken down. And there's this rhythmic fluctuation in these cyclins. Now the cyclins, as they're produced, will join with another protein whose level is constant in the cell called cyclin-dependent kinases. Once, and you can see that here. Here's our purple cyclin. Here's our cyclin-dependent kinase. So the purple molecules are fluctuating. The orange molecules stay relatively constant. But once these two join together, they form maturation promoting factor. Think of maturation promoting factor as mitosis promoting factor. In other words, it stimulates mitosis. And so the effect of this is because the cyclin concentrations cycle, so too does the MPF concentration. And you can see the timing of the M phase, mitosis, of the cell cycle correlates with when MPF levels are high. And then they drop, MPF levels climb, mitosis occurs again. They drop, MPF levels climb, mitosis occurs, and then they drop. And so you have this rhythmic cycle specific to each cell type. How does maturation promotion factor stimulate mitosis? Well, it breaks down the nuclear envelope. It triggers spindle formation, at least we suspect, and then it triggers the condensation of chromatin into chromosomes. And so again, like all those things that need to happen for mitosis to take place happen because of that MPF level, whose concentrations fluctuate based on this normal clock. Another way we regulate the cell cycle are with what are called checkpoints. Checkpoints are what are called go or no-go signals at specific points of the cell cycle, specifically G1, M, and G2. Think of them like little stoplights. We're kind of moving along, ready to divide, and then we hit this little checkpoint, and there's this little pause, and the cell kind of looks, okay, is everything looking good? And if so, all right, things are looking good, go ahead, and then we continue to divide. If, however, there's a problem, then that cell will immediately sort of peel away from cell division and either go back to G0 where it's not dividing, or some cases will just kill itself off through a process called apoptosis, which we'll talk about in a moment. So what are some of these stop and go signals that could be happening? Well, one are growth factors. Growth factors are just proteins that stimulate cell division. That's all they are. And so here's just an experiment where we take a little bit of human tissue and we pour them on a petri dish. And if we add growth factor, then they divide much more readily than if we do not add the growth factor. So they just stimulate cell division. Another stop or go signal is density-dependent inhibition. What that means is crowded cells will actually stop dividing. So if you put some cells into a petri dish, they will divide on their own automatically and spread out until they form a nice even layer across the bottom. If you scoop some out, they will divide and replace that missing part and then stop. This is called density-dependent inhibition. It's like, okay, I'm snug up against my neighbors. Okay, I won't divide. Well, certain cancer cells ignore that density-dependent inhibition. And so if you do the same thing with cancer cells, they just keep going and they'll grow and pile up into this big mass. So again, density-dependent kind of tells the cell, okay, there's enough of us, we're good, so we can stop dividing, and cancer cells ignore that. Another one is anchorage dependence. So a cell must be attached to an appropriate surface to divide. So again, if this cell wasn't attached to the surface of this petri dish in this case, it wouldn't divide. So again, that is to prevent a random cell from just dividing any old place if it gets lost in the body. In metastasis during cancer, again, the cancer cell is ignoring the anchorage dependence. So even though it's floating free through the bloodstream and attaches finds itself in some totally incorrect spot, it ignores anchorage dependence and divides anyway, causing a tumor in that new spot. So basically, if any of these little signals are not good and the cell detects a problem at these checkpoints, then, like I said, it will either stop dividing or it will undergo what is called apoptosis. Apoptosis means programmed cell death. And again, this should occur if a cell has damage that cannot be repaired. Unfortunately, apoptosis mechanisms can fail. And again, if the cell doesn't kill itself off, then it will continue to divide uncontrollably, which then would lead to cancer. So very important for cells to behave properly. And if they don't, then they need to kill themselves off. So with that, that's all I got. Thanks, everybody.